One,
yes,
two.
of course. What I
Next slide, please. Can you hear me at the back? Well, can you hear me at the back? Oh, now the microphone's got feedback. Next slide, please. Next slide, please. Please, can someone shut that door or stop the rock group in next door? And please, can someone kindly catch that swarm of bees? Next slide, please. I'm sure you can't make out the tracks, 'cause this has just come in by fax. I'm still expecting several more, so next slide, please. Next slide, please. Now this curve that goes straight down shows stimulation upside down or just a crack the wrong way round. I'm not quite sure. Next slide, please. Can you go 13 slides back? Or is it 16? I lose track. And no, I meant the left projector. Next slide, please. Next slide, please. Now this is looking rather blurred. Is it a plane or just a bird? I should have skipped that 7th whiskey. Next slide, please. Next slide, please. This may cause some slight confusion, 'cause it shows cold nuclear fusion. Now it's melting the projector. Next slide, please. Next slide, please. Have I been drinking too much liquor, or is that smoke there getting thicker? Can you find a new projector? Next slide, please. Next slide, please. It looks as if I'll have to shout. The fire alarm is winning out. Can you just try to pay attention? Next slide, please. Next slide, please. I don't think this slide can be seen. That fireman's hat obscures the screen. Can you just wait until I've finished? Next slide, please. Next slide, please. Now I'm feeling rather manic, but the audience is panic. Won't disturb my concentration. Next slide, please. Next slide, please. Now this looks like apoptosis. Can you turn off those fire hoses? 'Cause the water level's rising. Next slide, please. Next slide, please. I'd like to thank the organizers, those two penny-pinching misers, who would only guarantee my one-way fare. Last slide, please. I see the fireman's going frantic, so I'd best swim the Atlantic in despair. This is a nightmare experienced by most scientists. As I was walking one summer's morning, I made my way to the library in search of wisdom and education. I scanned the journals so eagerly. I picked up Nature and looked inside it, and lots of adverts I there did see for PCR and restriction enzymes, for cloning kits and HPLC. At last I tracked down the page of contents and scanned the titles so eagerly. There were quarks and quasars and plate tectonics, T-cell receptors and HIV. And as I browsed through the latest scandals, I sighed and yawned rather wearily, till there I saw my latest paper, and brightened up most immediately. I swelled with pride as I gazed upon it. What finer gels could you ever see? And every figure was as we'd planned it. Models of logic and clarity. But then I paused in deep contemplation. A nagging doubt stirred to trouble me. It was just 3 days since we'd sent the paper. There was no way this could be by me. The author's name and address were different. All other features were just the same. I gazed in horror at every figure. There was no doubt we were scooped again. I muttered oaths as I gazed upon it. I mopped the sweat from my fevered head. I'd have torn my hair, but that posed a problem. So I sighed and wished that I'd stayed in bed. I found a keyboard and started writing. A short erratum I sent straightway, correcting names and address of authors. And whistling softly I strolled away. That described one familiar sort of scientific nightmare. This describes another. It's not about reading the literature. Instead, it's about international travel. And in particular, it's about international airports and the stress that these impose on our lives. And for those of a nervous disposition, you'll be pleased to know that at least one airport caters for passengers of a nervous disposition, namely Narita Airport in Tokyo, where the instructions of how to find your way through the airport give a phone number at the bottom of the sheet, which you can dial for fright information. This song is about an airport nearer to home. One perk of being a scientist is that you get to go to exotic destinations like New Jersey and Glasgow. But there's one snag I'll tell you, for no matter where I go, it seems I always have to fly from London Heathrow. Up and away is where I long to go, but why, oh why do I have to fly from London Heathrow? In a ring round London Airport, there are traffic jams for miles and crowds of sweating bodies with more grimaces than smiles. You stand in line for endless hours that exhaust your temper's dwindling powers when you hear your flight will be delayed for only 19 hours. Up and away is where I long to go, but next time I fly I'll try to bypass London Heathrow. So I fly to Rome from Cambridge with one change in Amsterdam, and I park in the long-term bike sheds with no sign of traffic jams. At Amsterdam it's easy as I climb the moving stair and arrive at my connecting gate with loads of time to spare. Up and away is where I long to go, but why, oh why did I ever fly from London Heathrow? So I sit back and relax as my flight takes off on time, when I realize I've left my watch still set on English time. So I slowly realize that although the gate was right, because my watch was 1 hour slow, this can't be the right flight. So if this is not the 10:05, I wonder with alarm if this flight is for Bolivia or Dar es Salaam. So I listen very carefully, but the pilots don't say much, and everything they say in fluent double Dutch. Up and away is where I long to go, but why, oh why did I try to bypass London Heathrow? So I listen very carefully for any English version, as I wonder if I'm going to need Swahili, Greek or Persian. Then the pilot reassuringly suggests we'd like to know that very soon we're due to land in London Heathrow. Up and away is where I long to go, but why, oh why do I have to fly from London Heathrow? This song is an awful warning. Lord Nelson's renowned for Trafalgar. An apple helped Newton to fame. Ed Southern's great worth spread all over the earth because of the blot on his name. And sometimes I feel green with envy of those who reach fortune and fame. But if fame knocks at the door, then I won't be so sure.
So good afternoon, everyone. Today is the third day of our online Zellbiologie practical course. I am Stasyk, and today we start in the live stream with the experiment number 2, subcellular fractionation. In the beginning, I will explain to you a few important points, general points, and give you overview on the topics of today. Then we will come to your protocol book, which you already have, and we'll explain to you all the steps of subcellular fractionation procedure. And we will, at the end of this first part, we will have a video tutorial which we have recorded for you, that you can see all the details in video. If you have any questions, if you have comments, please write them to the chat. Also, at the end of each hour, we will have specifically some time, 5-10 minutes or so, for the questions, for your questions. So let's start. In this illustration, you can see how epithelial cell look like. You can see the nucleus, plasma membrane, which also make this nice brush border on apical side of the cell. Several organelles are depicted here, like for instance endoplasmic reticulum, Golgi, mitochondria, endosomes, lysosomes. So as you know from previous cell biology practicum, all the organelles have different functions in the cell. And the composition of the proteins and the lipids in the organelle define the function of organelle, but also size and shape of them. And this is important for the subcellular fractionation procedure I'm going to introduce to you today. The aim of the subcellular fractionation is to purify different organelles in subcellular fraction in as pure as possible and native state in the reproducible manner. In our practicum, we will purify two fractions. One is total membranes, which consist all the organelles, but excluding nucleus. And second fraction we will purify is cytosol. So this is the fluid part of the cell where all the organelles swim, so to say, in the simplified definition of the cytosol. So why do we need subcellular fractionation? And what are advantages of this approach? It's clearly the first step what we can do with in vitro cell lines, and in many cases it's essential step for downstream approaches, for instance, like proteomics. Why is it so? First of all, by identification and characterization of the components of the organelles of purified subcellular fractions and also multi-protein complexes, we can link function of these proteins and protein complexes to specific organelle. Second, we can enrich low-abundant proteins, among them signaling molecules which in general would escape identification. And this is illustrated here on the slide with the peak of iceberg, which represents the high abundant proteins, for instance, which we can see. But what we do not see is hidden there underwater. And these are lower abundant proteins which we can start to see and detect with the different downstream approaches. Important is the third point, that all the purified subcellular fractions and the endosomes will have reduced sample complexity. It will make possible identification of the components with the technology like, for instance, mass spectrometry. Of course, all the subcellular fractionation techniques which you use in a lab are flexible and adjustable. It depends on the scientific question you ask. And the last point I would like to make here is that subcellular fractionation can be combined with the functional analysis when we want to see basically not just the composition, but we want to see difference. We want to see how proteins, for instance, or lipids or metabolites are regulated, what is up or what is down regulated depending on the functional state of the organelle. For instance, starved versus stimulated versus the stimulated with growth factor, or the nutrients like, for instance, amino acids. Subcellular fractionation is a bit tricky method. We want to establish method which is reproducible, and it is not so easy because, first of all, the organelles are similar. The physical properties of different organelles are similar. They also look similar, like as you can see here in the electron microscopy picture. And because they are similar, they are difficult to separate and to purify. Second, different cell lines, many different cell lines we have in vitro, they are different in size and shape, and we have to adjust the protocols, so to say, individually and empirically. And it is clear that there is a big difference between cultured cell lines in vitro and the real tissues. So what we can do to disrupt cells and purify or extract specific molecules. We can use directly different chemical solvents to separate, for instance, water-soluble versus lipid, organic fractions. Many detergents are used in a lab to lyse cells, to prepare cell lysates. Additionally, we can use enzymes, for instance, lysozyme or chitinase, to solubilize the cell wall, for instance, or to get cells from the tissues. There are different physical approaches, like, for instance, the osmotic shock, when we can incubate the cells in the low ionic strength buffer, and the cells basically will swell and break, so that we can the inside can be released in the components can be released in the buffer. In the freezing thawing approach, cells will be broken by ice, by crystals of ice. There are many mechanical approaches, like, for instance, sonication, like French press, syringe and needle homogenization, which I will explain to you in the next slides. The first chemicals which we use, the most common chemicals which we use very often in the laboratories, are detergents. There are many different detergents of different chemical groups, and two of them are shown here as a Triton X100, for instance, and P40, just because they are the most common in the laboratories, but there are different, and also they differ mainly in the, so to say, lipid solubilization power. What is important that these two, Triton and P40, are rather cheap and available, but many detergents are expensive. And the most probably important that many of the detergents are not compatible with the downstream methods for the detection, identification, for instance, proteins, and they have to be removed in the additional steps. And detergents which are compatible with, for instance, mass spectrometry, are really very expensive. So in the French press approach, cells are placed in the stainless steel container, and they are disrupted by high pressure, which forces cells to go through the small hole. This is the way how cells are disrupted. Very common tools in the laboratories are sonicators, and the sonicator probe can be put directly in the cell suspension on ice, and the cells will be broken by ultrasound waves which come from the sonicator. So, very common approach. Another device, very popular, is the Down's homogenizer. When the cells are placed in the usually glass vessel, and they break between the glass vessel wall and the tight-fitting plunger when it goes downwards by rotating, and then upwards and downwards. So basically, they break here in between the wall and the plunger. And now we come to homogenization with syringe and the needle. This is the approach which we are going to use in our practical course, so stay focused. In this approach, cell suspension is pressed through the needle in a way that shear forces disrupt the cells, and the organelles and nuclei are released. What is important that needle size, especially the inner diameter size of the needle, it can be different. So we basically can empirically try and find the best size of the needle, which will homogenize the cell line of interest in the most efficient way, but also in most native and gentle way to preserve the integrity of the organelles. And now I would like to ask you the question. What do you think? How the diameter, the size, the inner diameter of a needle corresponds and correlates with the size of the nucleus in a cell? Do you think it should be smaller than nucleus, or the size of the nuclear diameter, or the bigger of nucleus? Think about how we homogenize. We will discuss cells and how we will discuss all the steps now. But what would you think would be the optimal? The size and ratio, the diameter of the needle to the diameter of the nucleus. And write it to the chat. I will check it time to time to see if there are answers. So, before homogenization, there are many important, sometimes simple things to consider before you start homogenization. And one of these the important points is the passage number of the cell. If you take cell line, for instance, we will work with HeLa cell line, as you know already. If you take them from the stock, from the liquid nitrogen, for instance, and the passage number is 5, and you keep the cell line for the experiment, and you come to the passage number, say, 35, this cell line might look different. Not just the simple passage number, but they might look different in the size, the morphology of the cells might be different, the cell-to-cell contact, confluency, and so on and so on. So when you start with homogenization procedure, pay attention on the passage number. Second is the confluence. Empirically, we know that the higher confluency of the cell line, cells in the culture, the better homogenization results are. Like something around 80% is the optimum. And it's not just because the more cells we have, the more final, so to say, product we get, the final organelles and subcellular fractions. But what is important when we homogenize cells, we actually break cell-cell interactions. So if the cells make tight contacts and they keep the shape, not like round balls, but they keep the shell like they were in the monolayer or they grown in cell culture, they are easier to homogenize. Right? So confluence is the second point. And very simple things, like for instance, is your shelf in the incubator leveled? For the subcellular fractionation, usually we need a little bit more cells. And for instance, if I take 15 centimeter dish, which is quite big, and your shelf in incubator is not leveled, I think it's clear that on one side, the cells might be already 100% dense confluency. On the other side, which is not level, it will be 40% confluency. So this is very important that the cells grow in the incubator, which is leveled. Now, this is the photo, the picture, how your bench look like before you start homogenization. So we perform everything. This is the most important for all procedure. We do everything strictly on ice. So you see crushed ice here on the ice box. We have a metal plate and a 10 centimeter cell culture dish, plastic cell culture dish, which is on the top of the metal plate. You can also add a little bit of water between the dish and the metal plate to be sure that the cells and the medium is cooled down very efficiently. Besides, everything what you're going to use, like tubes, like buffer, plastic Pasteur pipettes, and everything should be pre-cooled and organized in a way that you are ready for homogenization. So I will make a very short break to look at your answers. So Isabel said, I guess it should be equal to the nucleus. Moritz, if you want the nucleus to stay intact, if you, for example, want to look at the proteins it contains, the needle should be just a little bit bigger than the nucleus size. Elia said, I think it should be equal or a little bigger than the nucleus, but it depends on the approach of the experiment. Tommaso, I'd say that it depends of the organelle of interest. Yeah, I guess it should be a little bigger than the nucleus so that it can pass through the needle, but that cells are not able to pass. Basically, you are all correct. The correct answer I read already from several of you that the needle is a little bit bigger than a nucleus. So we want to homogenize, we want to break an open cell, but we want to keep the nucleus intact. Thanks a lot. These are good answers and very nice activity. So let's keep doing that. And to keep you also thinking and busy and follow me, on today's presentation, I will ask you now second question. And it's simple. So actually, why do we keep everything on ice? Why do we need all the procedure and all the components and the buffers and cells to have, let's say, ice 0, maybe +4, but basically it's around 0 degrees. Why it is important for subcellular fractionation? Also, please write your thoughts to the chat. Now, so we have our cells, rather confluent, 4 degrees, and how do we harvest cells? There you see again the photo. We scrape cells, and this cell scraper, or it's also called rubber policeman. Here you can see the rubber policeman which we use in our laboratory. So we sell biology department, so we have very specific cell scrapers, so to say self-made, but also in the Praktikum, the commercial one which we'll use will work nicely. So you basically scrape cells, and you will see it better on the video we have prepared and we will watch a bit later. And you collect them in the one side of the dish in the PBS buffer, right? So what is also important when we collect cells, that when we start homogenization, the density of the cells should be around 20–30% before we start homogenization with a needle. This is empirical. We just found out that this is, the results are better. And we have to verify before homogenization that there are no big aggregates, and we suspend these aggregates of cells before we start homogenization. Again, I will explain it to you in details on the video. Now we start the cells homogenization with the passages, with the pressing them through the needle. And after this homogenization and centrifugation, we will obtain the PNS, post-nuclear supernatant. When we use the optimal, let's say mild homogenization, we will get something like 50–60% cells nicely homogenized, and the rest, the cells will be unbroken, and also some of them will open, and we will obtain the nuclear pellet. So actually everything will be centrifuged in the pellet. So unbroken cells and the nuclei, and we will follow with this PNS supernatant. We control this homogenization procedure, and basically you will see on the video that this is about how you make how many strokes you make to the cells to this homogenization. I will explain to you later a little bit. And we can control this efficiency of homogenization under phase contrast microscopy. I don't think we will have a chance for all 29 of you in the Praktikum in September. Hopefully it will work, that everyone will check the homogenization efficiency, because of we have to be quite fast and efficient in this process, but we monitor this efficiency when we establish the method for each cell line and check the efficiency in phase contrast. And you can also use the trypan blue, what you have, staining, what you have used 2 days ago with Ilya Vietor when you were doing trypsinization and cell counting. So this also, all the experience you can use in this process. Now, you see the photo of how the syringe and the needle is positioned in the cell suspension, and then we start homogenization. So number of strokes, there are, I briefly mentioned, we have already established for the HeLa cells, that, and you can see it in your protocol book, that the 7 strokes, ups and downs, 7 homogenization steps is optimal for the HeLa cell line. So what happens if you make less, let's say 4 strokes? We can under-homogenize cells, and we can have still cytoplasmic aggregates, cytoskeletal proteins, and so on, where the organelles will be trapped, and they will go to the pellet after centrifugation. So this is the final results will not be optimal. So we will get too little total amount of organelle of interest, for instance. Even the worst situation is when we over-homogenize cells. So for instance, if you make, instead of 7 strokes, you make say 10 or 12 strokes. So we might come to the level of over-homogenization, that organelles will break and release their content, and basically the cytosolic fraction will be contaminated with intraluminal the proteins or the metabolites, And the really bad situation will happen if we break the lysosomes, and because, as you know, they have inside a lot of very active and hydrolases and other enzymes, and basically our sample can be degraded, so because of proteolysis. If we use, for instance, as we already agreed, that the size of the needle should be proper, a little bit bigger than the nucleus, but if you use too narrow, too small diameter needle, or we over-homogenize this number of strokes, we might get nuclear breakage, and then released DNA, of course, will be the source of the aggregates. So partially, I have answered already my question. Let's take a look again into the chat if we have answers. So that proteins or DNA molecules don't get degraded by proteases or DNases, which now float freely through the mixture. Yes, maybe to slow down unwanted reactions such as enzyme activity. Absolutely correct. Both are correct. So basically, especially now, since I mentioned this, the homogenization, that some of the, for instance, lysosomes can break, then we have to keep everything on ice, that even in certain percent of the lysosomes break, or the other organelles, that we keep the degradation as low, as controlled, as limited as possible. And of course, the enzymatic activity, when all the organelles are separated in the compartments and the proper enzymes in the proper place, this is okay. This is, so to say, in vivo situation. But when we break and open and everything in the solution, in the supernatant, then all the enzymes might work. So we really have to keep everything on ice. Thanks. That was good exercise. So let's proceed. We now, a few more general comments from my side, that certain cell lines are a little bit harder to homogenize because, for instance, after harvesting, after scraping them from the plastic dish, they might round up. Basically, they look like balls, right? The second situation could be that the nucleus size of the nucleus is very large. That basically, when you look at the under microscope, and you see that cell is, the volume is basically, the nucleus is almost the size of the cell. So these are really difficult to homogenize with needle and the syringe method. That's why for this we use hypotonic shock. So basically we incubate the cells for some defined time, 10 minutes for instance, in the low osmotic strength buffer, that cells will swell, right? And then when they are swollen, they are bigger size, then we start the homogenization. So this is one more comment. And some cells just basically grow in suspension. They are not adherent, and these cells also might be difficult to homogenize in this method. So in these cases, like I have already mentioned in the previous slide, Downs homogenizer can be used, and also the number of strokes or passages with the in the Downs homogenizer might need to be increased and can be quite high to get sufficient breakage and homogenization of the cells. Now, I would like to basically summarize what we are going to do in our practicum, and also what we will see. It's shown as in the figure, in the scheme at the page 11 of your protocol book, as we have already discussed. So we start with a cell culture. Possibly you will prepare cells yourself, or you will get it from us, from our technician prepare it. We scrape, as you know already, cells, harvest cells, and we homogenize with a syringe and needle, centrifuge, get a pellet, so nuclei and unbroken cells, and the postnuclear supernatant. We will take for the further centrifugation and at ultracentrifuge 100,000 G for 1 hour is standard approach to get pellet of total membranes. So it will consist of all the membrane organelles and the supernatant, which is cytosol. So everything what I try to show you now today was on the slides, on some details, on the photos and the pictures. Now I think it's time to start the video tutorial which we have prepared for you, that you can see everything in details. If you would have questions, if you have questions now, let me check chat. So far I don't see any question. Hopefully everything is clear, so that we start our video. And I will stop probably many times to point in something important what I think is. So this is the page from the first page of the experiment number 2, subcellular fractionation, from your practical book. And this explains again what I told you already, some details, and you can see the figure. What I told you now, basically this is the list of the equipment we need. It's simple method, it's cheap. We need a syringe and a needle. For HeLa cells we have already optimized before that we need 25G needle for the optimal homogenization, and you know why. Because of size of cells and nuclear size, and we need a cell scraper, an ice box, pipette, centrifuges, ultracentrifuge of course, and tubes. So everything has to be on ice. This is clear, we already agreed on that. Let's take a look how your bench look like for subcellular fractionation. So before you go and pick up your cells, everything is on ice. You see the metal plate, important. A bit of water is also good. The cell scraper here is on ice. I'm not sure if it's visible on the monitor, but there is a plastic transfer pipette here on the ice. The tubes, you label tube with your initial. So this should be my initial, so TS, it's mine. The second tube I will use as the balance for centrifugation. Since we will be in our practicum room more people, that we do not need to have a balance, we will just use all your tubes to centrifuge, but they have to be, of course, balanced. So what you can see also is PBS here on ice. There is a second tube which is labeled HB. Maybe someone of you already who read the protocol book. HB stands for homogenization buffer. And I didn't mention it so far, but this is the buffer in which we will actually perform homogenization. So we will have additional step to exchange the PBS in which we harvest cells, and the next step will be exchanging buffer from PBS, phosphide buffer, it's saline, the standard one, which we use in a cell culture. We will exchange to the HB, homogenization buffer. So this is the buffer which is isotonic and very close isotonicity and pH very close to what is the condition inside of the cell. So the organelles will feel kind of better, so to say, but for sure better that in PBS. You can check it in the protocol book. It contains sucrose and the buffer. So this is our homogenization buffer. We have a syringe, we have a needle, 25G, and the tubes. Of course, we will use the pipette boy. For pipetting, we need access to the pipettes and the tips. So I think we are ready to pick up cells. Let's go now to the cell culture. So this is the cell culture in our practical room. In the biocenter, we take our cells for homogenization. For the practical course, we will use HeLa cell line, 10 centimeter dish. Of course, you check the passage number. In this case, it was passage 26. It was done in the middle of April, and these are my initials, that you know that this is your cell line and your dish. So we take it, sorry, so I think I clicked on the wrong button. So let's now exercise a little bit, myself. So I took already cells, and I showed you already the HeLa, how it looked like, and now we go and place them on ice. So when cells are on ice, they cool down quite efficiently and fast, and the next step will be washing. So we want to remove the medium and wash cells with PBS. We wash cells twice with 10 milliliter of ice-cold PBS. We open the bottle, we put the plastic pipette which you have already used, so to say, virtually online 2 days ago with Ilya, and now we remove the medium to the plastic beaker. So this is the plastic beaker which we collect all the biological waste, all the media, all the washing buffer, all the components we use. Right? So we take 10 milliliters of PBS. Don't be super precise. If you take 9.5 or 10.5, it's absolutely fine. When you add the PBS to the cells, you see I added on the side carefully, not too slow, not too fast, that you do not wash your cells from the dish. Pour it out again to the plastic beaker with the biological waste. You take a second time of PBS and wash it second time. You shake dish nicely, that you kind of distribute PBS and wash out the medium from the cells, and pour it out and again to this waste. Now the third time you take 5 milliliter of PBS. Here try to be more precise, so the 5 exactly, and I'll explain you why it is important. 5 milliliters of PBS exactly. Now you add it, and now you do not throw it away, pour it out from the dish, but we start scraping. So now please pay attention how I do that. So this is our cell scraper. You put it in the 45 degrees angle with the sharp side of the rubber of the cell scraper to the cells, and you basically rotate the dish directly on the metal plate. Then in one move you can collect, say, 70 or so % of all the cells by keeping cells also on ice. So the one round, for instance, I started with this red position, and then there are basically 2 or 3 times you scrape and collect all the cells. So now I will try to show you. So probably now you see that cells which we scrape and harvest with this rubber policeman, you can see them here on the side of the Petri dish in the PBS buffer, and they are not single cells. This is not like a cell suspension. This is really the layer of the cell, and this is important. So basically you collect almost 100% of the cells in just a simple 3 or 4 moves, and they are all in this layer of the cell. This is how it should be done, and then we can transfer these cells from the dish to the centrifuge tube. So now you see again these layers. We take the tube, can open it before, not as I did, and with the plastic transfer pipette, you see we transfer the cells to centrifugation tube, 15 milliliter centrifugation tube. We call them also Falcon tubes. And we close the tube, make sure that you have it labeled properly, that you will find it, because if you have 29 tubes, then you will have to find your own tube, and we go to centrifuge. So this centrifuge, as you have seen, is by window, which is in the Inrheinstrasse in your Praktikum, in Inrhein, yeah, and the Praktikum room, and we start centrifugation. The first centrifugation step is 1000 rotation per minute. You can check protocol, 5 minutes, 4 degrees, and we start. So virtual centrifugation was fast. 5 minutes went like a second. And I will just now explain you very briefly why it was important, what I mentioned, that you add 5 milliliters. When we are in the Praktikum, many students, we do not measure the volumes to be sure that everything is balanced. So we just know that you follow protocol, and if you added 5 milliliters, we just balance all the tubes. If you need one balance, it's fine. And we know that they are 5 milliliter inside of each tube, so that we can centrifuge them. So this is important, so that you all add 5 milliliters of PBS for cell harvesting. So centrifugation is done, and we take the tubes from the centrifuge. Now we carefully aspirate and discard supernatant. We can do this with the blue tip and the 1 milliliter pipette, or we do the aspiration with aspiration pump, as you have done 2 days ago with Ilya Vietor in the cell culture room. So now you see the cell pellet, right? And be carefully remove the supernatant. Carefully that you do not disturb the cells. Don't try to, so I'll stop for a second, and don't try to remove the last microliters, right? So the 10, some 20 microliters is absolutely fine, because otherwise you will take some cells out of the tube. Let's see if there is no questions. So far so good. There are no questions in the chat, so we proceed. We aspirated or removed and discarded supernatant. And now this is the step of the exchanging buffer to HB, homogenization buffer. We take 2 milliliters of this ice-cold HB buffer, and we add it to the pellet, that entire pellet of the cell float up. It's a little bit difficult to see, if at all, on your monitors, but basically we add this buffer. We do not resuspend. We don't need to resuspend the cells. They will float because the PBase is lighter. The density is lighter than HB. And so this the entire pellet will float more or less on the surface, or the bit of the suspension, and we will perform the second centrifugation. What I do now, I prepare the second tube by adding 2 milliliters of HB buffer as the balance. But as I already mentioned, if all of us, we add 2 milliliters of HB buffer, then we know all of our 20 or 30 tubes will be balanced, and we can go for centrifugation, 10 minutes, 2,500 rotation per minute. The same centrifuge, we will balance all the tubes to the centrifuge, and we start centrifugation, make sure that all the parameters are correct. So we adjust the speed to the 2,500. Let's see if it's correct. Yes. So 10 minutes time centrifugation and 4 degrees, and we start. Now, 10 minutes, it's not really the time for the break, because we need to prepare the next step. And the next step is extremely important. We have to prepare needle and the syringe for homogenization. So they are already pre-cooled, but what we need to do is to take some volume of HB buffer that we know that inside there is a certain dead volume of all the needles and the syringes. So you connect tightly needle and syringe. We need to have an HB buffer, as you see inside of the needle and the syringe, and most important, to make sure that there are no bubbles. This is extremely important for the homogenization. We take some volume, let's say half milliliter or so. Then we put the needle and the upside down, and we shake. So we kick it with the fingers like that, and you see the bubbles. I hope you see that they are on the top. So when they are on the top, when we keep the needle vertically, and we remove by pressing the HB buffer from the syringe out to the paper towel. And you will see that you basically remove, by doing that, all the bubbles. And you remove all the HB buffer from the syringe. Make sure that there are no bubbles. And close back the needle and put it back on ice. So there is a question in chat. Isabel asked, can the weight of the cell pellet be neglected for the balancing in the centrifuge? Yes, absolutely. So this is we all get very similar, the confluency of the cell, and as you have seen, this is something like I would say 40-50 microliters of the pellet, and when you add 2 milliliters and all the others 5 milliliters and so on, the volume of the buffers, this contribution is very minor, and it will be actually very, very similar for all of us, all the students. I will just briefly mention now the bubbles problem here. If you have some bubbles here, you repeat this shaking, the pressing bubbles out from the syringe and needle as many times as possible. So usually it works in the first go, but sometimes when you do this for the first time, you might need to repeat a second time. When we have this centrifugation 10 minutes, it is more than enough time to make it properly and to try. you have enough HB buffer, and then you will remove all the bubbles. So it will work for you. I am sure it always works. What is important now, when you close this, the needle back to the plastic cover, don't do this too super tight. You don't need it, just to protect it and put it back on ice. Also be careful that you do not inject yourself. Also as important as please do not inject anything to your colleagues, to your group mate, and also your teachers. So an HB buffer, as we already agreed, we keep everything on ice, and to protect the possible sample degradation, we also add protease and phosphatase inhibitors. So this injection would not be really something good for us. That's why really be careful that you do not with these needles and the syringes, right? So let's proceed. We are ready with a needle for homogenization and syringe. We put it on ice, and now this is the pellet after centrifugation in HB buffer. What we again do, we carefully aspirate and discard the supernatant. Do you see the pellet? Yeah, this is the pellet. It stays as it was before, but now it is, the buffer is exchanged to HB, homogenization buffer. Carefully remove. I will probably stop it. You see the pellet. Remove the supernatant, but don't try to remove the last 5-10 microliters. This is absolutely okay. This is HB buffer. Now the next step, right? You have seen I took the pipette, because what we want to do, we need to resuspend the pellets in the 200 microliters of HB buffer for homogenization, right? And if you take a pipette, if you have to adjust what I'm doing now, adjust the volume. If you see directly here, 200 microliters, you put your tube with cells on ice. Yeah, always keep it on ice. And as you see, my tube of cells is on ice. Now, important thing is that you take your 200 microliters of a buffer in the blue tip and 1 milliliter pipette. This is very important. Now look carefully. We resuspend gently but efficiently. Cells 5-7 times or so, ups and downs. We need to resuspend these aggregates, the clumps. If you see visible aggregates, the homogenization will not be efficient, because they basically can clog the needle. We start homogenization. Everything is ready. We put a needle and the syringe completely down to the bottom of the tube. You see it here. And we start homogenization. We remember 7 strokes, but we start with taking suspension up. You do this gently. And again, critical point. Don't try to take the last 5-10 microliters. Why? Because there is a danger that you will take air. We don't really need the air bubbles, because the homogenization will be basically unpredicted. We don't know how homogenization will work if the air bubble will be there. So do this slowly, take it up and depress it down. Pressing down. It is not in the middle, let's say, suspension. This cell suspension from the needle will not go in the air somewhere. You will see, and I will try to focus it again with a few times stopping and restarting the movie. There is a very tight contact between the needle and the plastic wall. You see this conus, you see this plastic wall of the falcon tube. So now maybe you can see it better. So you see the needle is in a tight contact, somewhere in the level of maybe 0.5 milliliter or so, there is this, or even lower. So you know the needle has a cut. So basically it's a certain degree, a very nice cut and sharp ending of the needle. And exactly this side, which you maybe see, maybe it's for some of you depends on your monitor, you can see, but it goes into tight direct contact with the plastic of the tube. So this is the way, this is the place and time where the cells, after leaving the needle, get broken by shear forces and organelles and nuclei, as I have tried to explain you at the beginning of today's presentation, they get broken with the method of the needle syringe homogenization. So make sure that you do this properly. That's why I emphasize it already several times. So I think I was a little bit too fast. So where we were, let's see it again. On the movie, I think we were here, right? So this was homogenization. I explained you. So let's try again. Sorry. This movie is jumping a little bit. So this was homogenization. And no, sorry, it was a little bit too fast. So we start centrifugation, 3000, 10 minutes, 4 degrees. After homogenization, the same principle. So we put the balanced tubes, we start centrifugation. And what do we get after centrifugation? You can see here. So this milky supernatant is post-nuclear supernatant. And the pellet you have seen here was the pellet what we already discussed before. So this is this pellet of the unbroken cells and the nuclei. The next step what we do, we carefully take 40 microliters aliquot of the supernatant, of this milky post-nuclear supernatant, and transfer to the tube, which is labeled PNS. Right? 40 microliters according to the protocol are transferred to the tube, and the second aliquot will be 100 microliters. This is very important. 100 microliters, very carefully. Look at the ending of the tip. Don't touch the cells here. So you take 100 microliters of the post-nuclear supernatant and transfer it to this special tube, which is labeled P100. This is special ultracentrifugation tube, and we will now go for the ultracentrifugation with this P100 tube. This is a rotor. This is special rotor for the shape of like EPIs tubes labeled P100, and these tubes survive this high speed. So normal Eppendorf tubes will break, they will not stand the ultracentrifugation. Also rotor, we put everything balanced, the second tube or all the 12 samples will fit into this rotor. Rotor is high-tech technology that can survive this rotor, for instance, 150,000 Gs. That's why everything has to be really balanced. I put it inside and I click on the button as a safety button that everything is correct in ultracentrifuge. So what we proceed, we check a rotor. I will stop in the next moment, 100,000, 1 hour, 4 degrees set, and we start. Some centrifuges, so I will stop for a second. So TLA55 is a rotor which we use. Some centrifuge recognizes it automatically, or you have to check it yourself. So we were centrifuging properly. Also the beginning was a little bit in the different rotor. So now we did everything correctly. Centrifugation is over. We turn off vacuum and we take the rotor and our samples from the centrifuge. So press again the button, take the rotor out from the centrifuge, and let's take a look carefully what we got. Often students on the practicum ask me, I don't see the pellet, I don't have a pellet. No worries, pellet is inside of the tube. It is just very small and it might be difficult to see. Let's try to see the pellet what we got. So this is our tube 100, P100, and this is the pellet. I stopped it. So this is the pellet, and this is your pellet of total membranes. The supernatant is cytosol. So always try to find it on the side, right, after centrifugation. So we got the pellet of total membrane fractions, and what we do next, we transfer 40 microliters of the supernatant, 40 microliters to the tube, normal Eppendorf, which is labeled S So we take it from here, right, after centrifugation, and we transfer it to the tube, which is labeled S100. This, yeah, exactly, and this is our cytosol. 40 microliters, the same volume as we have P and S. Right? So again, carefully that you do not disturb the palate of the total membranes after ultracentrifugation. And the rest of supernatant we collect and discard. We don't need it. Take it, you don't see the pellet, but this basically the pellet is left in the tube and the supernatant is collected and discarded. So now we got all the samples after this ultracentrifugation, original postnuclear supernatant, S100, which is cytosol, and P100, which is pellet 100,000 G, is all the organelles, membrane organelles, and the total membrane fraction, what we call it. So this was the video, and this is the end of the video. If you have any questions, let's see, let me check in the chat. So if something was not clear, we have enough time that I can repeat part of the video tutorial or explain you something later after the break. So please write your questions. I will also check the chat during the break. So far, I think everything seemed to be clear. That's why we are perfectly in time. So let's have 10 minutes break, and we will meet exactly in 10 minutes for the second part of our live stream today. See you soon.
If you listen, I'll try to explain. For no matter what I do, I fear I'm destined to be known as the man who coined that ghastly term, molecular chaperone. One day I found a new protein that acts in an unusual way. It helps other structures assemble by keeping their ligands at bay. It doesn't bring partners together, but rather it holds them apart to protect their virginity at any salinity by playing a chaperone's part. This protein I called nucleoplasmin, and I wanted to say how it acts, and molecular chaperone seemed a good term to accommodate most of the facts. For this term I had planned just a quick one-night stand, then I'd carelessly throw it away. But I ought to have known that no true chaperone could allow me to act in this way. Now, no matter what I do, I fear I'm certain to be known as the man who coined that ghastly term, molecular chaperone. I'd failed to allow for John Ellis, a true British bulldog of old, With a shotgun he said, "To this term you'll be wed," and he welcomed it into the fold. I never thought that one cute concept could cause me such anguish and pain, but to increase my groaning he coined chaperonin and used it again and again. So no matter what I do, I fear I'm certain to be known as the man who coined that ghastly term, molecular chaperone. How can I escape from this problem and banish this term from your heads? I need a distraction to divert your minds and leave a new image instead. So I need a new image to help me escape. So I might have to take up fraud, arson, or rape, or even descend to recording this tape to silence those desperate groans. I'm divorcing those damn chaperones. This is politically and ecologically correct. It consists almost entirely of recycled garbage. Namely, some of my previous songs. I rejigged several songs together to form this one, rather to the astonishment of a class of 350 undergraduates to whom I have to deliver the last block of lectures before the vacation. And spirits are always running rather high in the last lecture immediately before the vacation. So to try to keep a lid on their enthusiasm, I wrote this song with the intention of singing them the chorus if mayhem broke out, and refusing to sing any more until I got to the end of the lecture. 4 years went by without them disrupting the lecture. So in the 5th year, I finally gave up and sang it to them anyway. The lecture series takes place in a lecture theatre called the Babbage, named after Sir Charles Babbage, who invented a mechanical forerunner of the computer. If you're feeling like a cabbage after too long in the Babbage, and your mind is full of Mendel clams and yaks, then it's time for celebration, 'cause here comes a new vacation. So forget about those lectures and relax. If you're feeling like a zombie after hearing too much Pombi, and your mind is full of yeasts and frogs and flies, you've seen each kinase from the zoo, lots of phosphatases too, and you're seeing cycling cells before your eyes. If you're feeling deep neurosis as he raves about mitosis, with the gleam of microtubules in his eyes, then I think it might be best to try G0 for a rest, and to borrow someone else's college prize. If you're feeling like a cabbage after too long in the Babbage, and your mind is full of Mendel clams and yaks, then it's time for celebration, 'cause here comes a new vacation. So forget about those lectures and relax. If the seats are getting harder and the clock seems to have stopped, and your lower half is slowly getting cramped, then if you try to reach the table where that damn projector's propped, maybe you can reach the wire and fuse the lamp. But if you try to have the last word in a hail of paper planes, so the room becomes an aerial traffic jam, then you might like to remember as you join in all the games, I can always get my own back in exams. If you're feeling like a cabbage after too long in the Babbage, and your mind is full of Mendel clams and yaks, then it's time for celebration, 'cause here comes a new vacation. So forget about those lectures and relax. Go and have your photo taken in the market in Tangiers, or maybe go and see the sights of Greece. But the only sights you'll see are 100 other students, and you saw all them the year before in Nice. As for me, I'll stay at home, no longer will I roam. I don't care what the staff and students say. I'll buy a sunray lamp and a set of foreign stamps, send myself a reprint request every day. 'Cause I'm feeling like a cabbage after too long in the Babbage, and my mind is full of Mendel clams and yaks. But it's time for celebration, 'cause here comes a new vacation. So forget about those lectures and relax. This song was found by a colleague of my wife's on the internet, but not in the form of a song, in the form of an article on the internet, which consists entirely of quotations of things that children have written in history and English lessons. I've taken the punchlines from those things and built a verse around each punchline.
So welcome back to the second part of today's online stream on the second experiment, subcellular fractionation. I see in the chat that we have many questions. So let's start with the first one. I see Moritz asked, what does 25G mean in the context of the needle? So the needle have these standardized, so they of course are used in the clinic, so the size of the needle is expressed in the gauge, so this G stands for the gauge, and this doesn't directly correspond to the size of the, let's say, diameter. It's a standardized values, and basically I can tell you just for your general feeling that, for instance, 25G would roughly correspond to 0.25 millimeter of the inner diameter, but these gauge numbers they reflect the outer and inner diameter of the needle. So these are kind of standardized numbers for the needle size. And the second thing I can mention that the lower G number, the bigger, so to say, the diameter of the needles are. And we also use different, like 27 and 25, for instance, we use in the laboratory for different cell lines, as an example. Second question from Isabel, if you do an experiment where you want to examine, for example, DNA or proteins that are inside the nucleus, do you also use the needle fractionation method, but with a smaller diameter than the nucleus? No. So from the fractions, so the procedure, what we have done, we do exactly the same, follow the same protocol as what we have done together with you now, today, virtually, and the pellet, after first centrifugation, you remember when we got the post-nuclear supernatant, so pellet contained the nuclei and unbroken cells. So this pellet we can resuspend and perform additional centrifugation step that we will collect the pellets from these original pellet what we have got. So we can collect very pure nuclear pellet from the pellet what we have obtained with you, and these pure or purified, very much enriched nuclei, then we proceed for the further studies. So we can study basically everything what is inside of the nucleus, right? So we can do proteomics of nuclear proteins identification, we can identify metabolites, we can follow some possibly nucleic acid research. So this is how we purify nuclei. Now, Catherine asked, point 10 in the protocol, for what reason do you exactly 40 microliters before ultracentrifugation? So this is the good question. Now I will explain to you in details, because this is the post-nuclear supernatant aliquot, which we will use for the further detection and determination of protein concentration. So now, today, in the second part of the stream, I will explain it to you. And this will be also the fraction for the further analysis with electrophoresis and Western blotting. So the volumes, you will see why we need exact numbers. So the microliters, 40 microliters, it's something what we can take as aliquot if we start with 200. Yasmin asked, how does the shear force with the needle and the syringe exactly work? Is the needle only pressed onto one side of the wall? So, well, cells, when they leave the needle, and basically when they go in the needle and they are in a close contact, in a tight contact with the plastic wall of the tube. This is the moment how the shear forces, right? So it goes into contact between the needle and the plastic when the cells get broken. You cannot really try to find the best golden place for the homogenization in the tube, but it's more or less on the side of this plastic, the conus, so the angle of the bottom of the falcon tube. And important is that you see this cut ending of the needle, that you come into close contact to the plastic wall. You will see it soon when we start homogenization. Hopefully we will have a chance to perform also a real practicum together with you a little bit later in September. So I hope I answered all the questions, and we can proceed with the second part of today's presentation. I think we don't have to go back to the movie, and I will proceed with the next slide. This is a very general slide for you, just to give you impression that basically actually all of the subcellular fractions and all of the organelles, even in the case if you cannot purify them in the, let's say, 100% purity, but there are approaches that we can separate them sufficiently enough to identify their proteome, metabolome, for instance, lipidomics also, and at the end, with careful and sensitive analysis, with instruments like here, it is a photo of the mass spectrometers which we have, and we use, for instance, here in the Biocenter campus. These are just an example of the detections and some mass spectra for you. Basically, at the end, we can make and construct something which is a cell map. And hopefully you will be interested after this course, and then you can also search yourself some literature and also for your homework to look what is the current state with the so-called cell map and how much we know about the organelles and the organelle proteomics and so on. And for this, I will just show you a little example, rather short example, how we do this in our laboratory, how we use very similar the protocol with what we discussed with you in the first part of today's presentation, and how we apply this protocol and subcellular fractionation to purify endosomes. Right? So you know, we start with cell culture, we harvest cells, and already here we can add the lysis buffer, standard lysis buffer to the pellet, what we got after first centrifugation step, to lyse cells and get total cell lysates, that we basically see all the components what is in the cell, so total proteome, for instance. Alternatively, what we have done with you, we went for the homogenization, and we separated into two fractions. One was the pellet of nuclei and unbroken cells, and the second supernatant, post-nuclear supernatant. What we have done with you is ultracentrifugation. We took the 100 microliters of the post-nuclear supernatant, and after 1 hour centrifugation at 100,000 G, we have got two fractions, cytosol S100 and the total membranes B100. for any of the fractions as well as any of the purified organelles, we can further go for the fractionations, like one is shown here, to purify, separate peripheral from integral membrane proteins. We go in the lab also for the Western blot analysis of the specific marker. I will come back to this in the next slide. And we also can prepare samples for downstream identification with the, for instance, proteomics, lipidomics, or metabolomics technologies. So what we do to purify endosomes from post-nuclear supernatant, we use gradient ultracentrifugation in the sucrose gradient. One of them is shown here as a continuous sucrose gradient. When we put post-nuclear supernatant on the top of the gradient, sucrose gradient from 10 to 40% sucrose, all of the organelles which are in the post-nuclear supernatant can be separated according to their density. I mentioned at the beginning of my presentation the proteins and lipids define the shape and size of the organelles, but what is important is the ratio between the lipids and the protein content define the density of the organelles. So the organelles can be separated as, let's say, light organelles like late endosomes, lysosomes will be distributed on the top of such organelles, and all the others more heavier like early endosomes and then ER, Golgi, and so on, so-called heavy membrane fraction, they're distributed throughout all this sucrose gradient. We collect many samples from such a sucrose gradient after ultracentrifugation, which usually takes long, like 20 hours or so, and we analyze them. We analyze markers of interest with Western blotting. This we will discuss in details tomorrow. And when we know the position of organelles of interest, for instance, late endosomes, we can design step gradients or discontinuous gradient. For instance, it's shown here that we have 4-step gradient like 8, 25, 35, 42%. Sucrose that we can separate late endosomes and early endosomes as an example in more quantitative amounts if you want to study the late and early endosomes. Or if you go for the 3-step gradients, 8 and 35 and 42, the mixture of all the endosomes will concentrate in the interface between 8 and 35% sucrose. So this is how we purify endosomes in our laboratory for different projects. And the example of such a endosomal purification I would like to show you on this slide. So for this we use this step gradient, 8 and 35% sucrose, and purified crude endosomes. So what is crude endosome? CE abbreviation stands here. This is the mixture of all the endosomes, recycling endosomes, late endosomes, early endosomes, lysosomes also. And what we can do by doing Western blot analysis, we can detect the markers of specific endosomes, like, for instance, LAMP1 for the late endosomes transferrin receptor, for the recycling and early endosomes, RAP5 for early endosomes, RAP7 for late, P14 protein, marker of the late endosomes. Also some signaling molecules like kinases, ERK is shown here. So they all concentrated in the very narrow part of this step gradient, which correspond to the interface between 8 and 35% sucrose, as you can see here in the Western blot. Alternatively, when we want to separate, for instance, late and early endosomes in analytical and in the nice way, when we use a continuous gradient, as I mentioned, so late endosomes are lighter, they have higher lipid content, and they come as a peak here, probably number 8 and 9, 7 to 9, the peak of the late endosomes. So the markers, like LAMP1, you already know from the presentation from Ilya. Yesterday, immunofluorescence, LAMP1, you had the RAP7 in the late endosomes, and the P14, another marker protein. They are here concentrated and co-migrate in the continuous sucrose gradient, the same time where the transferrin receptor and the RAP5 as the markers of early endosomes, for instance, they are separated and they're peaking at fraction number 14, 15. So this is the example how we analyze our samples of the purified endosomes by Western blotting. So far no further questions on the chat. And I will show you the second example of the downstream technology how we analyze the purified endosomes or subcellular fraction in this case. Maybe not many of you know the technology which is called two-dimensional gel electrophoresis, where we can label purified protein samples with different fluorescent dyes. Like for instance here, post-nuclear supernatant is labeled in the blue, cytosol in the green, and the one of the subcellular fractions, specifically peripheral membrane proteins, are labeled in red. So all the samples can be mixed in one sample and analyzed in two-dimensional gels. The principle of the method, I will just mention it very briefly, it's clearly out of the scope of today's presentation, but we can separate the proteins in the according to their isoelectric point. That would be this horizontal separation, and molecular weight on the vertical, let's say here, directions. So what you can clearly see, and I would probably come back to my original, one of the first introductory slides when I mentioned that we can see some low abundant organelle or subcellular fraction specific proteins. So you can see the red, which correspond to the peripheral membrane proteins, are very enriched in these fractions, and many of these red labeled membrane proteins we would not be able to see on the level of total cell lysates, or often even on the post-nuclear supernatant. So you see clearly the effect of the enrichment of the proteins, and you see clearly the cytosolic, for instance, green, many of the green proteins which we can enrich in specific purified subcellular fraction. And you remember this peak of iceberg. So we can really dig deeper now and to see very low abundant also signaling molecules and the signaling protein complexes. So coming back to our protocol book, to our practicum, we were on the level that we have homogenized with syringe our cells. We prepared PNS and prepared cytosol and total membranes, two subcellular fractions and original post-nuclear supernatant. So what we do now, what we are going to do, then next step is to analyze markers by electrophoresis and Western blotting analysis. And this will be topic of tomorrow's presentation. We will use two markers already mentioned, and you know already, the transferrin receptor, the membrane protein, so marker of total membrane fraction, and you know about the tubulin cytoskeletal protein component of microtubules, which were already nicely presented by Mariana Araujo in the first introductory slide, all the mechanism. And now you see we are also going to use it as a marker for our subcellular fractionation, as a marker of cytosol. So, as I mentioned, of course, specific markers are required to follow all this procedure. I already briefly mentioned some of the proteins which we can use as a markers, and maybe to define what is defined as a marker. So this should be specific protein which is specifically localized to one or another subcellular fraction or the organelle. So we know that this is protein, for instance, which is localized to lysosomes, right? So this will be the marker. And often such a proteins also have a specific function in this organelle of interest. And some of such markers are mentioned here on the slide, like, for instance, RAP5 and transferrin receptors, the markers of early endosomes. At the same time, the transferrin receptor can be recycled back to the plasma membrane, to the cell surface. That's why it's also the marker of the recycling endosomes. And early endosomal antigen 1, it was also mentioned before, also, and some RAPs, RAP4 and 11, also marker of recycling endosomes. And as I have mentioned already, RAP7, and you know also from immunofluorescence from the previous presentation by Ilya, that we use the RAP7 as the marker of late endosomes. LAMP1 also would be good marker to mention also here. And we can use also some of the enzymes. So the enzymes which are enriched in the endosome, late endosomes, or the lysosomes. Besides speaking about the enzymes, there is a very interesting approach that we can feed cells with enzymes like HRP, horseradish peroxidase, for instance, and we can follow basically all the endocytosis pathway from the internalization via vesicles to early endosomes, to late endosomes, and down to the lysosomes, for instance, for the degradation. So basically, we can follow the time course. Usually it is in about, let's say, 10, 20 minutes, and then in a level of 20, 40 minutes, it reaches late endosomes and lysosomes. So basically, we can also follow our markers together with the enzyme, which was endocytose, and combine this information to know where we are if you want to study endocytosis, for instance, by subcellular fractionation. But before we go for the analysis, what is important to do? We want to know what we got, how much we got a protein. Is it sufficient for downstream analysis? Besides, we have to normalize all the samples of one of another way. In any situation, all the biological experiments, and subcellular fractionation is one of them. We perform in a lab, we should know how much protein we get. There are many different approaches how to determine the protein concentrations in a biological sample, and I'm going to introduce to you the Bradford Assay, which is very common, and that is the reason why I want to talk about that. Besides that, this is nice, reliable, and very commonly used in laboratories, many different kind of laboratories. The method to determine the protein concentration in the sample. I put here that we refer to original publication, which was published by Bradford in 1976. What is interesting, this is the third most cited paper in the scientific literature, which speaks about how important this discovery was, that we still, so many years passed, and we still use this method which was described so many years ago, and that is one of the most cited papers. I put a question to you now. If you know already, you can answer. Which publication in scientific literature is the second one in this ranking? So what is the second most cited paper? If you know, you can put it in chat. Maybe someone had this question, asked this question before. If not, then you can leave it just for your general interest, and you can check it later, today, for instance. The method is simple, and simple is beauty. The Kumasi G250 dye, some of you might know Kumasi to stain the protein. Be careful, because for staining of proteins, maybe you had other courses for electrophoresis before. You use most probably different Kumasi, which is called R250 Kumasi dye. And this could be another question for you, just who is interested. Don't have to put it in the chat, but you can check it later. When you have time, what is the difference between the Kumasi G250 and R250? One is used for this Bradford protein determination, and another one is common for the protein staining, although this one also can be used for the protein staining in the gel electrophoresis. So this dye binds to the proteins in acidic medium, and what we can see basically immediately the changing in the color. It changes the color from the brown to the blue. And this blue we can measure with the readers or the cuvettes, spectrophotometers. So we will do it in 96 well plate format, and we can read it and measure the absorbance at 595 nanometers. To know exactly, because we will get certain value, certain number, so to say, when we read it in 96 well plate reader. But of course, what we need to have is the standard curve. So the known protein concentrations we will prepare during the Praktikum, and we will perform the same reaction, so incubation with the Kumasi dye or the Bradford reagent. And since we know the protein concentration or amounts in the sample, we will make a standard curve. And for the standard, as a standard protein, very common, almost exclusively, is used BSA, bovine serum albumin, to prepare the standard curve. And you will do this during the Praktikum, so that's in your protocol. Now we again refer to your protocol book. All of you will prepare 200 microliters here of the albumin standards, dilutions in the water. So you will have to calculate how much you should take from the stock. You all get, I believe it should be 1 milligram per milliliter, and that you will prepare your standard dilutions, like for instance 200, 100, 50, 25, and so on. It should be milligram per milliliter. So sorry, it was just copy pasting from the protocol book to the presentation. It's not recognized. So simple things I would like to mention here. It happens very often that standard curve for a few students from the whole group looks very strange, and we found out that the reason was vortexing. So whatever solutions you prepare, standard curve, standard dilutions, any buffers, everything, of course you have to mix it properly. You will have in the your benches vortexes, so you have to vortex your Eppendorf tubes when you prepare the standard dilutions of the BSA. And then basically there is the pipetting scheme. This is an example. You don't really have to do this exactly like it is written here, but we will use 96 well plate. And as an example of a pipetting is shown for you here. So the 40 microliters of this BSA standards, you pipette in the well of 96 well plate as a blank. So the water, 40 microliters, the fixed and known amount of the BSA, as it's here, for instance, 0.5 micrograms in 40 microliters, 1 microgram, 2 micrograms, 4 micrograms, 8 micrograms, as an example. Of course, this is absolutely flexible. The method is quite linear in the broad range of the concentrations, but we expect, and we know from our experience, that we most probably will be in this range of the protein amounts, what we will be able, and concentrations to measure. So how we can optimize also the protein measuring here. So as I mentioned, we will determine protein in post-nuclear supernatant only. We take a water, 38 microliters, and pipette it in the empty the wells of 96 well plate, and we add 2 microliters of the PNS, post-nuclear supernatant, sample and mix well. So here it's shown the duplicates. Usually duplicates are sufficient. Sometimes it's important to make it triplicate, and this will be our sample. Of course, you can see immediately that in this way how we prepare biological sample, we can of course take 3 microliters or we can take 5 microliters, right? So this is a little bit of experience that 2 microliters most probably will be okay, so to say optimal for the protein determination, but it gives you a big flexibility, because even in this 96 well plate format you can take 40 microliters of the sample for the measuring, but you can keep in mind that in certain situations you have to consider buffer. So in our case it will be HB buffer as a blank, so not the water but the buffer, what you use for whole protocol. And then when we prepare all the samples in duplicates, we add Bradford reagent, 200 microliters, and I'll show you on the movie how we add it. So you add 200 microliters, incubate 10 minutes at room temperature. So basically reaction is very fast. After 3-5 minutes you see already the color, and it's quite stable up to 1 hour. You don't have to run to the plate reader to read your results, because it will be quite stable at least between 5 minutes and 30 minutes. It's absolutely fine, and we will measure the absorbance at 595 nanometers on the plate reader, and we will see it now very soon on the video we have prepared for you. Now, Elias answered the question, and obviously it is the second most cited paper, and the title is Cleavage of Structural Proteins During the Assembly of Head of the Bacteriophage T4 9070. The answer is formally correct. This is the title of the paper, and you still can take a look carefully what was described in this paper, and we will, I think, start tomorrow. I will mention this paper, what method was described, and why it is on the second position of the most cited paper, why it is so popular, what this method is about. Title is absolutely correct and citation is correct. But let's take a look, spend more time and find what method was described in this paper. Okay, but so now we look at the video for the protein.
I work next door to this guy.
Really? Yeah. So Professor Huber just said that he was working next door in Geneva to this person who is the author of this paper. So take a look again in this publication, what Elias put here in the citation, and you will know whom we are talking about. This is very interesting, and I will explain to you all the details why it is so important tomorrow. So today we proceed with the video tutorial for the protein determination by Bradford Assay. It will be rather short. So you see, I will start with explanation. So this is a commercial Coomassie plus protein assay reagent. So basically we still call it Bradford reagent or Coomassie reagent. I know some of my colleagues also still use the classical Bradford reagent. Now, since, as you now know, it was published and developed, described so many years ago, 1976, that companies try to optimize it, commercialize this reagent, that it's a little bit more stable and less sensitive to contaminations and the presence of, for instance, detergents, right? So that's, we also use, this is the reason that we use the commercial Coomassie plus reagent, but the classical Bradford reagent will work nicely also. So we have this Bradford reagent. We take aliquot. So you calculate how many milliliters of this reagent you need. You plan it in advance. You take certain aliquot. So I took some, probably 15 milliliters or so here for my experiment. And this is already, so this was already the prepared 96 well plate with our samples, right? You see it on the bench. So this is ready. I didn't show you how to pipette 40 microliters into 96 well plate. You will manage it. And this is ready to measure. You all individually will get, or the group will work in the Praktikum in the groups. You will get such 96 well plate, and you will pipette the standard BSA dilutions and your samples as we agreed. So 38 microliters of water and 2 microliters of post-nuclear supernatant. And well, I didn't triplicate, but I think we will also can do this in duplicates. It's sufficient. And then now the next step that we take 200 microliters of the Bradford reagent, 200 as you can see, with a multi-channel pipette. So in this multi-channel there are 8 channels. So we take yellow tips now and carefully take the Bradford reagent. This is sometimes difficult when you do this for the first time, because when you take it tips from the box, that you take it not, so to say, strong enough or equally press one left and right side, and then it can happen that in the, let's say, in one side, you see that the reagent doesn't come to the same level and the other tips. Of course, we control that. Yeah? So that's if you see something is wrong, you blow it out back and you throw out the tips and you take new ones. So you will have a chance and play with the tips how to make it properly. But this is an example how I did it some time ago when we were recording the video tutorial for you. So you take 200 microliters in 8 multi-channel the pipette. It worked nicely, and you add it to your samples. Now, you add it carefully and you resuspend your samples. 3 times is sufficient. And you throw out tips. You always take a fresh tip. Press firmly and nicely that you are sure that you will take the reagent nicely, and you again add to your samples and mix it. Now, you see that in my case it worked nicely, maybe because I did it already many times. What is important that when you mix it and when you add reagent, that you do not introduce the bubbles. So the killing bubbles in each well, especially if you have 96 well plate, take time, and this is really not necessary. But the bubbles, again, the same or similar situation that we didn't, we try to avoid the bubbles, or we avoid bubbles in the needles and this syringe for the homogenization. Similar situation is here. If you will see the bubbles in the 96 well plate, you would have to remove it, because the values which you read in 96 well plate reader will not be correct, right? So the mixing of the reagent with the sample is important step. It looks simple, but it's important, and you will have a chance to exercise in that. So now we add reagent, and we come to the 96 well plate reader, which is in your practicum room. According to protocol, we incubate the plate for 10 minutes at room temperature. And I stopped the movie now for the one specific reason. You see, it's actually appeared after, almost immediately after a minute or so, that you see the color changes. So here on the top, this is our blank, the water, and as I mentioned, it's like brownish, light brown color of the sample where there are no proteins. And then the standard curve in triplicate here, so we increase the concentration from lower to higher, and you see this is like a dark blue in the highest concentration of your sample. So sample of interest, like post-nuclear supernatant, 2 microliters we took in 40 microliters total sample volume, is here on the bottom. So this is the row H here on the 96 well plate. And you see a certain color. What is the beauty of this method? That actually even without a reader, you can take a look at your sample, besides the paying attention on the bubbles, hopefully absence of the bubbles, you can see that the color intensity would correspond, well, probably not to this row, right? And not to this, but something close to that one, maybe something in between. So you can just look at the color and say, well, my concentration is in this range. Cannot tell you now exactly, I don't remember by heart what was the concentration of the protein, this in standard curve, but you can estimate it quite well just visually, right? So let's proceed where we are. So this is your, again, maybe closer look. You can appreciate the color brown to the blue and your sample, and no bubbles, luckily. And I think we should wait 10 minutes, not now, but 10 minutes on your timer. Now we have 10 minutes from this moment. We start our plate reader. So this is the view from the monitor or computer connected to the 96 well plate reader from your practicum room. And this is already window opened that you see photometrical measurement. Basically, you can adjust quite many different parameters, which wells you want to read, how you read the spectrum or the specific wavelengths. Do you want to shake, temperature, incubation, so on and so on. In our case, it's simple. We don't need to control the temperature. Also, we do not need to shake in this case. We just select the wavelengths. So what you pay attention now is 595 nanometers, and we will read whole 96 well plate. So basically what we need to do is just start. Important is to be sure to make sure that this is 595 nanometers for the blue color of the Bradford reagent. So basically we are ready. That's what I told you already, some of the checking parameters. And we will start reading our results in 96 plate reader. So we take our well and put it prepared, ready to read. I will stop and make a short pause here for the one simple reason. Might sound stupid, but in some cases the 96 well plate are also with the leads. So don't forget to remove the lead before you insert it into the reader for measuring your results. So there are also different methods when you have to incubate your sample for a longer time, and then you use a lead. So make sure that you insert the 96 well plate without the lead. And now we are ready. We just press start. And so we press start here, 595. Yeah, now it's very fast. The reading takes like a 15 seconds or so, and we have numbers. They will appear very fast. So now the plate is inside, and these 2 seconds, 3 seconds were sufficient to get the numbers. So they are a little bit not sharp to see on the video, but basically you have seen on all the positions of the video here of the 96 well plate. I'll try to maybe stop for at the beginning of this measuring that you will see certain numbers. No, they are not sharp enough, but basically there are numbers. We don't need to discuss, but here maybe you can see it's 0.22 or something, and then it's increased to 0.4 or some this order of the values which correspond to increased. Yeah, so now you see it better. It again jumps. So 200, 300, 400, and so on. That's the higher concentration in the standard curve, then you get higher values. So I just paid attention in the chat, and Magdalena answered that this was, we were talking about the Lemley and the gel electrophoresis, and she even mentioned 213,000 citations of this paper. Exactly, this is the second best, well, best, not really, but this you cannot compare scientific publication this way, but this most cited paper, this is from the beginning of the publications and checking citation indexes. So Lemley, gel electrophoresis, second most cited paper, absolutely correct. And interestingly, tomorrow this will be topic for our tomorrow's new live stream and the second part of the subcellular fractionation experiment number 2. So at the end, this was already the end. Okay, it jumps because it's the end. So you see the 96 well played out from the reader, and we are done. So we get the values in the computer. There are two options. We can print our results if you want to analyze and proceed with our experiments the same day in the Praktikum. And we can do this in the way that in your protocol book. So this what you have in the page. Let's see what page it is. It's page 14. You have exactly the same, the millimeter paper, so to say. You can make a graph. It sounds like old-fashioned, but this is very useful so that you can prepare your standard curve, so your values in the based on the BSA standard, and the optical density is at 595 nanometers. And you can make a standard curve directly in your protocol book. So here I suggest you put the micrograms of protein, so the amount of the protein which was in the well, which produce and give us absorbance at 595. And so there will be certain standard curve. Not necessarily it will be absolutely linear. It might come to a certain plateau, but you can make it directly in your protocol book. You can also use microgram per microliters. It's basically up to you. Most important that you know and understand what you're doing. Alternatively, if you don't do this directly in the day of Praktikum, you will get all of your results, your own results, as Excel file with all your raw values in duplicates or in the triplicates, depends how we will do that. That you can make your graph standard curve in the Excel file. I'm absolutely sure that you're all familiar with Excel, how to make such a standard curve. And this is an example of my curve I have done for my experiments some time ago. And here are the BSA in the micrograms, for instance, and optical density. And this is the curve I got. So you can also ask software to show you the formula, like for instance, the formula for such a averaged, like linear, because it's rather a linear standard curve here. And the formula you can use to calculate the protein amount and at the end the protein concentration in your post-nuclear supernatant sample of your individual, what you will prepare yourself. Here as an example, I give you the 3 different samples of postnuclear supernatants from the last year protocol, from the last year practicum. 3 different students prepared PNS 1, 2, and 3. We took 2 microliters, as we already discussed and agreed, of the sample, and these are optical densities of these duplicates, sample 1 and 2, for the 3 students. So now I will ask you the question, what volume of the PNS1, PNS2, PNS3 samples is to take 30 microgram of proteins for Western blotting? So the question most probably will stay with you till tomorrow. I think you also have these values numbers in your uploaded PDF file, which you have access to in Elias. So think what volume, basically now we are not yet on the level of electrophoresis and Western blot, but we ask a question, do we have sufficient, is the sample what you have purified of postnuclear supernatant enough to load 30 microgram of protein for the Western blot analysis? So I think I will, if someone did it already, you can put it in the chat, otherwise we will discuss it tomorrow. We probably will start with this slide tomorrow, the part 2 of the experiment subcellular fractionation, which will be about downstream analysis, SDS page, so protein electrophoresis, and the Western blotting analysis. I think I am close to the end of my presentation. And yes, so this is my last slide when I would like to thank you for your attention, for your active participation with the questions and discussion in the chat. We will stay for the next, say, 10-15 minutes in the chat, that you can still write your questions if you have. And you can also write emails if the questions will come later, that I will answer your questions tomorrow, and we will start next day presentation with your questions. Thanks again, and see you tomorrow. Ciao.
Richard Lederer, so wherever you are, Richard Lederer, thank you. From the mouths of babes and sucklings, many pearls of wisdom fall, and I'm glad to say a school teacher has tried to catch them all. And the verses of this song I'll sing obey one simple rule. They're based on things the children wrote in history class at school. Now the mummies lived long times ago, and Egypt was their nation. They cultivated deserts by using irritation. And the ancient Greek philosophers of history were the bedrock, but Socrates, he met his end by an overdose of wedlock. The next lot was called Romans, in verses, books, and songs, because they never stayed around in one place very long. Next came the Middle Ages, with jousting and wild games. King Arthur's knights were chivalry, but Alfred conquered dames. When Elizabeth the First was queen, they thought that she'd go far. She exposed herself before her troops, and they all shouted hurrah. John Milton was a writer, for Paradise Lost he's famed, but when one day his wife died, he wrote Paradise Regained. Newton was a scientist, he invented gravity. There's more of it in autumn, so the apples fall from trees. Pasteur found a cure for rabbis and other strange diseases. Charles Darwin was a naturist who wrote The Organ of the Species. And finally, Sir Francis Drake, that well-known sailing skipper, he circumcised the whole wide world with 100-foot-long clipper. Those of you who heard the previous tape that we made in here will think you know this song. You're wrong. Little boxes, little boxes, little boxes in the genome. Little boxes, little boxes, little boxes all the same. There's a cat box, there's a tatter box, there's a Homea box, and there's a Pribner box, and they're all made out of ATGC. They all look just the same. So the scientists all clone them and run gels to read the sequences, and they all find little boxes all the same. And you might think that these boxes are boring with just 4 nucleotides. But remember little phosphates, 'cause they all look just the same. Little phosphates, little phosphates, little phosphates on the proteins, little phosphates, little phosphates, little phosphates all the same. They're on serine, they're on threonine, they're on tyrosine, and they're on histidine, and they form part of every nucleotide, and they all look just the same. And for every little phosphate, there's a kinase and there's a phosphatase, and they all try to move the phosphates so they won't all look the same. So the scientists all map them and use analogs to trap them. Then they all publish little motifs, and they all look just the same. Then the scientists read the papers and all rush to find the kinases, and they find one, then they find one, then they find one yet again. Then they name them and they blame them for life, death, and almost everything. Then they all publish little cascades, and they all look just the same. Now map kinase, 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 phosphorylates map kinase, kinase, and that phosphorylates map kinase, and that's where the trouble starts. 'Cause for every little kinase, there's a whole zoo of protein phosphatases, and they all take away the phosphates, so we're right back at the start. Then receptors in the membrane start the whole kinase cascade signaling. Then they all modulate the signals, little signals all the same. And the signals reach the nucleus and phosphorylate transcription factors, and they all bind to little boxes just the same. Little boxes, little boxes, little boxes in the genome. Little boxes, little boxes, little boxes all the same. There's a cat box, there's a tatter box, there's a Homea box, and there's a Pribner box, and they're all made of blasted phosphate, and they all look just the same. You'll all realize at once that the contents of this next song are so far-fetched that they couldn't possibly be true. Sadly, they are. It's a song that's dedicated to a noble British institution called British Rail. And the first 3 verses consist entirely of excuses for why the train network was seriously disrupted. And these were the actual excuses that British Rail offered. The remainder of the song consists of 2 true stories that happened to me while travelling from Cambridge. So this is a token of my esteem to British Rail. Dear passenger, there's something you should know. That when it snowed last night, it snowed the wrong kind of snow. And with this sprinkling on the ground, our high-tech wheels will not go round. They'll slide and they'll slip and they'll get no grip, and so our trains won't go. It's the wrong kind of ticket, it's the wrong kind of snow, the wrong kind of temperature, that's why our trains won't go. Now the gentle breeze that disturbed the trees brought the leaves all a-tumbling down, and they all fell back across the track, all slippery and brown. And with these leaves upon the ground, our high-tech wheels will not go round. They'll slide and they'll slip and they'll get no grip, and so our trains won't go. It's the wrong kind of ticket, it's the wrong kind of snow, the wrong kind of temperature, that's why our trains won't go. Dear passenger, we've had a dreadful fright. The temperature dropped rather low, it even froze last night. And with their mechanisms froze, the doors won't open and won't close, so our reputation grows. 3 cheers for British Snail. It's the wrong kind of ticket, it's the wrong kind of snow, the wrong kind of temperature, that's why our trains won't go. Now I journeyed off to Kent one day for a meeting that was there, and I bought a return ticket paying twice the single fare, and on the return